Okay, ladies and gentlemen, let's get started. I apologize. On Monday, some of you know I have a lab that goes to 425 upstairs, and sometimes it takes a couple extra minutes to get down here to get started with you guys at 4:30. So what's going on today? I'm really glad to see this many people here. I'm really very excited.
Why?
Well, because today's content's not on the exam, and that sometimes means that people all just stay home. So I'm just really glad that you're here. Okay, let me just say thank you. Okay. So today we're going to be finishing up, not finishing up, but continuing our discussion of Chapter 3 on cells and organelles and all of that good stuff. And I wanted to see if there are any questions. Take a few minutes. Are there any questions related to the exam? On Wednesday, we'll be meeting here, not here, right? 1304, Wednesday. You'll have with you a Scantron 884-E, the big 8.5 by 11 green double-sided form. They're the cheapest ones in the bookstore. They're 19 cents a piece. The half ones are 40 cents. I don't understand why, right? But we're using the big one, the cheaper one, little savings on you. And all you need is a pencil and have your ID with you in case I see you and I'm like, I don't know who you are. I'll check it. And come in prepared. That exam, again, will cover chapters 1 and 2 and all the material from Lab 1. So you've had that material in your hands for a little while now. Hopefully you're feeling comfortable with it. And is it tonight or tomorrow night the quiz is due? Tomorrow night. Okay. Okay. So tomorrow night, I heard consensus. Tomorrow night, by midnight, make sure you have completed the online quiz that's related to the content for Chapter 1. And that should make you feel more comfortable with material. Okay. What else? Anything else? Vocabulary will be 1 through 16. I'm not going to do any vocab today. I'm not going to go on and do 17 through 20. I'm just going to save that for another mind. We don't need to worry about that today. Yes, Michelle.
Okay, like the pre-quiz or the quiz that we take tonight, the Zoom tonight, tomorrow night, is that kind of like summing up everything that's probably? I mean, I know they're gonna be.
It's not. I wouldn't call it. What's the word I'm looking for? It's not as comprehensive, right? Because there's going to be 100 questions on the exam, right? But it's all the ideas. So if you do well in that quiz, then that's helping you study. It is open book. It's open resource. It's really another opportunity for you to get a good grade. And then your exam, that's worth 10 points. Your exam will be worth 80 points. So really between the quiz and the exam, you get 90 points. Yeah.
The online quiz, is it like you get 10 points for doing it? Or
No. The online quiz is graded for how accurate you are.
Okay.
Now the homework assignments, right? Those do as well as you can, right? But when the quiz time comes, yep, this is right is right and wrong is wrong, and your grade will be reflected in that. Okay? So the quiz is real.
Everybody is done with it?
After everyone's done with it, you'll see you'll see it. Okay? Other questions?
I just have a question regarding
Chapter
chapter 2.
2.
Before you go into chapter 3, or can you just mention this? Is this will be on the quiz about the matching of the DNA and RNA? Like, I can't pronounce it. Like adenine.
Yeah, adenine so I briefly told you for chapter 2, we'll talk more about it in this coming chapter. But for DNA, I told you that it's double-stranded, and that A's match with T's, and C's match with G's. That much you need to understand, but that's it. Pretty much for DNA. DNA is double-stranded, whereas RNA is single-stranded. And the other difference is that RNA has in it uracil, whereas DNA has in it T's, thymine. So those couple distinctions, right, that I told you just as an introduction about nucleic acids. Okay. Any other things to talk about from chapter 2 to clarify? Something comes to mind, let me know. Yes, ma'am.
Okay, with the cell, we will be labeling it.
Not on the exam.
Not on the exam.
The cell is chapter 3. So everything I did last time and what I'm doing today is for exam number 2. The content here is chapter 1, atom to atom hierarchy body systems. Chapter 2, chemistry, atoms, molecules, macromolecules, what's a lipid, a protein, a nucleic acid, and lab 1, which was body cavities, directional terms, serous membranes, that content, plus vocab 1 through 16. That's exam 1. About 100 questions. largely multiple choice. There'll be a few fill in the blank. Vocab will be fill in the blank. Maybe a couple other simple questions, but you'll find it mostly to be multiple choice. Yes?
If we started the exam, we can still exit out of it and it is.
The quiz. Yeah, the quiz is not timed,
It is
right?
our spot.
So you can come and go.
Okay.
The quiz you're doing that's due tomorrow night, you can come and go.
Just once?
Say it again?
Ask how many times?
No, you only take it once,
Okay.
but you can come and go. You can go in, spend 20 minutes and leave and come back, and yeah, it's mobile that way. Okay,
Wednesday, all we're doing is the exam?
and Wednesday, all you're doing is the exam.
Okay.
Yep, the exam will take we have the entire hour and a half period to take the exam. Most of it will be done in about an hour. It's the shortest exam of the year, but most of it will be done, but you'll have an hour and a half. I don't think anyone will stress over time on exam one. Very rare.
As long as you.
Okay. Okay. So I'm not going to do any vocab today, and I'm just going to flash through these slides. Last time we talked about the cell. We talked about the characteristics of cells and the size of them and the functions of them, and you labeled the different organelles. And we spent a lot of time talking about the plasma membrane, right? And how some molecules can go in and some molecules cannot go in. Passive versus active transport. Endocytosis, exocytosis, and then I began introducing to you the organelles, and we started talking about the rough ER and the smooth ER, and we got to the Golgi, and I think that's about where we got, right? Yes. So let's back up just for a second. So looking at this slide, what page is this on in your notes? 40 something, 44? Okay, so I just said that really fast. Anything that you can think of in there, right? I'm not going to spend a lot of time on it because it's not on the task, and you can ask questions about it as it starts to sink in the next week or so. But here we are, we're inside the cell. We're looking at the Golgi apparatus. The Golgi apparatus is this folded series of membranes, and this folded series of membranes is packaging what is coming in on the receiving side from the ER. So the ER is making the proteins, the ER is making the fatty acids, all that stuff is going to get packaged at the Golgi, and some of it will be sent out as individual molecules, but much of it will be packaged in these little vesicles. Some of these vesicles are packaged with specific sets of enzymes, specific sets of molecules, and one of those specific little vacuoles or vesicles is a lysosome. We'll talk about those in a moment. Some of the other molecules would be packaged for release. Right? So there's an exocytotic event happening across the bottom. Maybe these are some sort of hormone or molecule that's being released from the cell. So you see them being exocytosed. And this picture also reminds us that the cell membrane is continuous, right? And that it's always ebbing and flowing. As things are exiting and entering the cell, the membrane is ebbing and flowing with it. The membrane is not just a sac, a bag around the cell that stays the same all the time. So we see that things are moving from the ER to the Golgi and then out to the rest of the cell. There's another type of small vesicle made, and I'm just going to draw it here, and this is going to be called a peroxisome. Okay, I just put a P in it for now. But we're going to be talking about lysosomes and peroxisomes. They are individual separate organelles that have a very particular job. So as I mentioned, these two vesicles, the lysosome and the peroxisome, are made by the Golgi. Right? It's a separate little set of enzymes that are packaged together to do a particular job. Okay. So lysosomes first. Last time I described to you how the cell can phagocytose a large particle. And that large thing is now inside the cell. Well, now what? Right? So the cell has to break down that big thing that it brought into the cell. If it were a white blood cell, it could be bringing in an entire microbe, an entire bacterium. If it's another cell, it could just be grabbing big chunks of nutrients, if you will, from the environment. So that's what's shown here. In fact, this one is showing what? This is not showing phagocytosis. This is showing not a big chunk coming in, but this is showing cellular slurping. I'll go back. This is showing penocytosis, right, where a little invagination occurs. So regardless, the molecules that were on the outside of the cell, right, this is the extracellular fluid out here, are now inside the cell. They're locked inside the cell. The cell can do nothing with those unless, look what's coming off the Golgi. Here's the Golgi. The Golgi has made a lysosome. That lysosome has within it a special set of enzymes that can pretty much break down everything. There are proteases to break down proteins, there are lipases to break down lipids, there are molecules to break down starches and everything else you could ever imagine. Remember, these are like little bubbles. They got little double membrane around them. And so what will happen is that the lysosome will bleb with the vesicle that came in from the penocytosis. So now this content will blend. And those enzymes in the lysosome will now break down these outside molecules down into their basic pieces and parts. So it would break proteins all the way down to amino acids. It would break nucleic acids all the way down to their nucleotides. It would break down sugars into their individual simple sugar molecules. And now those molecules can be used by the cell. So now those basic molecules can be used to build up something else. So we see that the lysosome is very, very important to the cells that are constantly bringing things in. And again, they are filled with enzymes that help to break things down. Now, lysosomes are also important in another cellular process that I've not yet described, and that is apoptosis. The word across the bottom. Apoptosis is cellular death, programmed cellular death. Last time I think I alluded to the fact that we have 75 trillion cells in our body. And I may have said that every second we're making 25 million new cells. I may not have said that, but regardless, we are making a huge number of cells every second, which means we're also losing 25 million cells every second. Okay? There are cells in our body that are supposed to live a short while and then are supposed to die. That programmed cellular death that's occurring in our body is apoptosis. And lysosomes are important in this because when the cell begins to degrade, the lysosome begins to basically eat away from the inside, right, and destroy that cell. So the lysosome is also important for cells to undergo normal cell death called apoptosis. Now you might be thinking here saying, back up. Another thing you can think about for the lysosome is that they're like the cellular garbage men. They're picking up all the debris. They're picking up dying organelles. They're picking up, they're helping to break down cells as they die. So they're really, really important. And if the lysosome is defective, then as you can imagine, it's like when New York City, when they go on strike and they don't pick up the garbage, right? It starts to stink really fast, and a lot of garbage gets piled up on the streets. And so lysosomes are very, very important for cellular function. The other little vesicle that I want to introduce you to are peroxisomes. They're like a little cousin. They contain a set of enzymes referred to as oxidases. Okay? Oxidases. And what these oxidase enzymes do is they oxidize molecules. Now, oxidizing oftentimes will kill, they will destroy molecules. If something becomes oxidized, oftentimes if it's a bacterium, it will be killed. If it's a molecule, it will be degraded. So it's similar, right? That we're breaking things down with the peroxisome. Just a different set of enzymes that do it. Now, when you hear about peroxisomes, they also are detoxifying, right? So you find a lot of peroxisomes in liver cells. What else did we say we found a lot of in liver cells? Because they detoxify and break things down, we also would see a lot of smooth ER, right? A lot of smooth ER in a liver cell, and a lot of peroxisomes because it also is breaking things down. So the liver, big time in the peroxisome world. Now, you might think, well, this seems awfully academic. These organelles, I really don't care much about them. I want to convince you, though, that for every organelle in our body has to be working, every cell has to be working at full function in order for us to have normal life. So here's an example of a disease that is caused by a faulty lysosome. It's Tay-Sachs disease. You may have heard of it. It's not an uncommon disease. It runs more in families from Eastern European Jewish ancestry. but it also is not that uncommon in Irish Americans, so it's not just a Jewish disease, if you will. It is transmitted by autosomal recessive. I'm not going to go into the genetics of this, but what this means is that two individuals can be totally normal, and their children, if both of mom and dad carry this gene, it's possible that their kids have this Tay-Sachs disease, and it kind of pops out of nowhere, right? So two very normal, healthy parents could have a child with this disease. What happens is because the lysosome is defective, it doesn't have all of the enzymes necessary to do its proper jobs, certain materials start to build up inside the cell. The garbage builds up. And one of the molecules that builds up slowly starts to shut down the nervous system. And there's actually more than one version of Tay-Sachs. There's 40 different enzymes inside the lysosome. And so if any one of those is messed up, you could have a Tay-Sachs-like disease. This disease, no cure, right? Death by age 4 to 5. And really not much we can do about these kids at this point. Now, the big thing for Tay-Sachs is that in Israel, it's not happening in this country, but in Israel, families always, the parents always get tested for this, right? Are they both carriers? So the incidence of Tay-Sachs has gone down in Israel because Jewish families realize this disease is higher in their genome, and they are active about testing to see if they're both carriers. And then they make other decisions about having kids. But in this country, we don't routinely check for Tay-Sachs. Okay, so that's the disease of the lysosome, right? Just to give you an idea. And then the peroxisome, you can't tell them apart. I would never ask you, is that little bleb in the cell a lysosome or is that a peroxisome? I can't, you can't tell them apart by looking at them. But there's another disease. Maybe you saw this movie, Lorenzo's Oil. It's probably a good 10, 12 years old now. And this was a movie about a parent and a parent's quest. Their little boy has adrenal leukodystrophy. And this disease is a peroxisome problem. So in his peroxisome, he did not have the right enzymes. And as a result, similar to Tay-Sachs, there's a buildup of fatty acid and fatty molecules that start shutting down the nervous system. And eventually these kids lose their reflexes, they become blind, and again, death by age 10 typically. This one hits boys more than girls because of the way it's transmitted. I'm not going to get into the X-linked versus the autosomal, but this one affects boys more. In the movie, it's kind of a good movie, kind of a tearjerker, but at the end, they solve the problem. And they discover that by modifying his diet, they can minimize the cumulative effects of the fats, and he can live a long, normal life if he avoids or has certain oils in his diet. So it's a fun little story, but I don't think anywhere in the movie did they say peroxisomes, right? But this is a peroxisome issue. So I'm just trying to drive home the idea that every organelle must be working fully for your cells to work, and that there are plenty of diseases out there where it's the organelles that are not working well.
Can you just explain what's the, like, compare and contrast, like, I don't understand, they both do the same thing, but what's the...
A lysosome has a certain set of enzymes that break things down. A peroxisome has a different set of enzymes. We'll go there, right? So lysosomes break things down typically. The oxidases oxidize and essentially break things down as well. I'm not going to get into that too craziness, right? Just know that there's two separate little sets of enzymes, and when they're not working well, disease is the result.
So they're both like garbage men though?
In a way, you could both think of them as garbage men. Yeah, yeah. Okay, so the last organelle that I want to talk about related to, remember these were the membranous organelles, the ones that have a membrane around them, is the mitochondria. Now, mitochondria, you could have hundreds, thousands of them in every cell. They're considered the powerhouses of your cell, and they are going to be the end of your metabolism. So when you take in sugar and you take in oxygen, and out of it we get ATP, right? That whole idea of cellular respiration that I kind of went over very, very quickly. The mitochondria are hugely involved with that process. And because of the mitochondria, it's here that we get those ATP, right? So without mitochondria working well, you do not get the energy that you need. And there are lots of diseases where the mitochondria are sick. I don't have an example of those. But you can imagine if your mitochondria are not working efficiently, you would not have sufficient energy to do your daily work. And these individuals with these mitochondrial diseases start going blind. And they start having other issues because over time, their brain's not getting the energy it needs, and other parts of their body are not, the muscles are breaking down. They're not getting the energy that they need. So, no, it's not MS, but I should put a slide in. I don't have one right now, but there's a whole slew of diseases related to bad mitochondria, but not MS. MS is a different story. Looking at the mitochondria, these little bean-shaped, they're about the size of a bacterium. So that will give you an idea. When you look at the cartoon in the bottom left, you can see that there are 3 little mitochondria in that slide. So mitochondria, about the same size as a bacterium, and you can see now the relationship of a eukaryotic cell, our cells, right, human cell, compared to a bacterial cell. There's a couple of unique things about this. There's a double membrane. Now, when I say cell membrane, you're thinking, oh, there's 2 layers of phospholipids, right? That's one layer. There's actually a true double layer. Where else do we see a true double layer? Around the nucleus. Remember, the nucleus truly had a double membrane all the way around it, except there were some gaps, right? Those nuclear pores. So the membrane has a true double layer of membrane, and so too do the mitochondria. All I really want you to know is that these are the organelle that produce the ATP.
Is there always 3 or just?
Oh no, 100. There could be hundreds.
Oh, okay.
That cartoon just shows 3 for simplicity.
Okay.
Now, imagine what kind of cells would have a lot of mitochondria? Would have a greater number? Muscle, right? Muscle needs a lot of energy. So you better believe that muscle cells are going to have a lot of mitochondria. And then other cells, maybe your skin cells, your epithelial cells, wouldn't have as many. Right? Again, yeah, so the ratio, the percentage or the number of these organelles within a cell has a lot to do with what the cell normally is doing. Now, this is just that overview slide again. We're not going to get into glycolysis, but the idea again, you take in some sort of energy, glucose. Glucose will undergo glycolysis, glycolysis, the breakdown of sugar. And as long as there's oxygen around, it'll go to the mitochondria, and in the mitochondria is where we'll get all those ATPs. Okay, that's all I want you to know. Okay, so we'll get our ATPs from the mitochondria. This is also where we get those CO2s. And we blow off that CO2 as gas. If there is no oxygen around, we don't use the mitochondria, and we instead do anaerobic respiration. And you may have heard of this. This is where you get lactic acid buildup. So when your muscles are working really, really hard and there's not enough oxygen going to your muscles and you start getting that burn in your muscles, you don't have enough oxygen going there, you start getting a lactic acid buildup, okay? And your muscles also are only getting 2 ATPs. So they're not getting very much energy when there's no oxygen around. So we need to have lots of oxygen in order to get lots of ATP. And the mitochondria is a part of that. And that's all I'm going to say. I'm just not going to go into the full story. We could spend 5, 6 lectures talking about how this works, and that's not the purpose here. So the mitochondria makes the energy. So overall, we would call all of the chemical reactions happening in your body your metabolism, right? Metabolism. And what are the 2 words we can use to break down or to separate metabolism? Catabolic and anabolic, right? So we have catabolic and anabolic. And catabolic were what? Breaking down, right? And anabolic were building up. Absolutely. So glycolysis would be what kind of reaction? Breaking down sugar. It'd be part of your catabolism, right? Breaking down sugar would be part of your catabolic reactions. And again, like I said in that picture, as long as there's oxygen around, your mitochondria will help you make 36 to 38 ATPs. If there is no oxygen, anaerobic, the absence of oxygen, then you're only going to get a couple of ATPs. So the cell, to do the most work, must have lots and lots of oxygen. Okay, that's just a quick view of what the mitochondria is doing for you. Then we've got the few non-membranous organelles, the organelles that do not have membrane around them, and this is going to include your ribosomes. This is going to include the proteins inside your cells, like the cytoskeletal proteins. This will include also the centrosome and the centrioles, the cilia, the flagella, and the microvilli. So we'll look at each of these briefly These are all considered organelles of the cell as well. So we'll start with ribosomes. We've seen them. Where did we see ribosomes? They were studied on the rough ER. Now, there are also plenty of ribosomes just floating out in the cytoplasm on their own. So they're either stuck to the rough ER or they're just floating around, right? And their job is to make proteins. This is their job. They do protein synthesis. They synthesize proteins. Now, a ribosome, and we'll look at them a little bit more later, but they're kind of small. They're really very small. You really can't see them with the light microscope. They come in two parts, a big part and a small part. That's easy. Large subunit, small subunit. And it looks like little tinker toys, right? So, not to memorize this, but there's a small part and a big part. And together they make the full ribosome. In this picture, we're seeing them dotted along all these membranes. So this is rough ER, right? And we can see those little ribosomes kind of studying along the endoplasmic reticulum. And then the picture in the bottom also reminds us that these little ribosomes can be freely floating. So they're either stuck to or freely floating in the cytoplasm. They're all about making proteins. That's all I'm going to say for now. We'll come back to the ribosomes a little bit more later in this chapter. Then there is a cytoskeleton, what gives the cell its structure, right? Its skeleton. And there's 3 different types of proteins found inside a cell that collectively give the cell its shape. So far in lab, pre-lab 2, and everything you've done so far, we've had you label some sort of spherical cell, right? But as we move through this unit, you're going to realize that some cells are very long and skinny, some cells, there's just lots of different shapes to cells. So they're not all round balls, okay? So these different proteins are going to help give cells their unique shapes. 3 varieties. Microtubules, as the name suggests, micro, small. It's almost like you're looking through a paper towel holder, the roll, right? There's a tube-like arrangement. And we're going to see that cilia and flagella are made of, or are partially made of, microtubules. Microfilaments, micro, small, filaments. Where until a few years ago would you find filaments in your home?
Light bulbs.
Light bulbs, right? Those little tungsten filament, right? Before the government got involved. So that little thin wire. So when you hear about microfilaments, think of very, very thin, wiry-like proteins. We're going to see microfilaments again when we get to muscle, and we talk about actin and myosin. Those are filament proteins. More about that later. And then finally, there are intermediate filaments. Guess what? They're kind of in between, size-wise. So you got these microtubules, you have these microfilaments, and then you have the intermediate filaments, and they're all doing something slightly different to give the cell its shape. Look at the top on the right. That's a microfilament. This is just a cartoon of it. That's the skinniest one. Then the intermediate, and then finally the biggest of all, and you literally would be able to look through it and see light on the other side, a tubule-like structure. They're all over the cell, right? They're giving the cell shape. They're giving the cell connections with other cells in the neighborhood. We're also going to see that during mitosis, when the chromosomes are being pulled apart, that's going to be microtubules, right? Mitotic spindles are microtubules. How cells move and migrate have a lot to do with these different proteins inside the cell. I would never have you label it and say, oh, that's a microtubule versus that's a microfilament. They're all simply cytoskeletal proteins, right? They just come in 3 flavors. Then there are cilia. When a cell has cilia, there are many of them. Okay, so there can be many, many, many cilia per cell. They're going to be long. They're going to be visible under the light microscope. And we're going to find cilia in places like the respiratory tract. Along the trachea, you would find cilia. And the cilia would be beating, and their job would be to help keep dust particles and bacteria from getting down into the lung. Then there's flagella. I really should say flagellum, right? Flagella is plural. I really should put um on the end. There's only one, and it's only going to be found in the human on sperm. Now, sometimes sperm have 2 flagella, and they just kind of go in circles. They don't know where to go. They're not very good, right? So they're mutated sperm, right? But normal sperm, ones that can reach the goal, have just 1 flagellum. And then there are microvilli. micro, very small, villi, finger-like extensions, tiny, tiny, tiny. Too small to see under the light microscope. But along the membrane of a cell, there could be these little tiny projections. And they're going to be there to give increased surface area. So just like a filter, when we get to the digestive system in a couple of weeks in lab, actually next week in histology lab, we'll be talking about this. But inside, along the small intestine, there are going to be cells with microvilli. And those microvilli are going to help increase the surface area. On the inside where the food is traveling through the tube. Okay? And because of all these microvilli, they're not like cilia that are waving to move things, but they're there to increase the surface area. So it makes all these little infoldings. And the greater the area of the gut, the more nutrients that can be absorbed at any one time. Okay? So that's the idea of microvilli. And we'll see them next week. Now here is a cartoon representation of this. In the upper, this would be cells. These cells are tall cells, and you can see up here some cilia. Those cilia are visible under the light microscope. If you looked at them, it would look like this over here on the right. Seagrass, or some sort of, summer wheat in Kansas, all sweeping and moving in the same direction with the current. On the bottom, there is a flagellum, single flagellum on a sperm. Okay? And then lastly, microvilli. I don't even have a picture of microvilli because you can't see them under the microscope. They would just barely give the cell a little bit more surface area. Again, those are all specializations of cells. You can also consider them organelles. Then we've got the centriole. Okay? In the cartoon of the cell, there was a centriole / centrosome, and that's kind of confusing. 2 centrioles make a centrosome. So centriole, O-L-E on the end, means little, small. So you've got 1 centriole, 2 centrioles. Together, 2 centrioles make a centrosome. Okay? And the only place we're really going to see these in mitosis, when the cell is actually dividing, what goes to the north and south pole, each of the centrioles will go one to the north pole and one to the south pole. And just like Spider-Man, will spit out from it the microtubules that will reach out and grab those chromosomes and pull them to each side of the cell during anaphase. Okay. So the centrioles are critical for cell division. Without them, a cell cannot divide. Let me tell you a little aside. Neurons don't have centrioles. So the cells that make up your nervous tissue, that make up your brain and your spinal cord, do not have centrioles. That means that they cannot divide or replace themselves, which is why when someone has brain damage or spinal cord damage, waiting is not going to make a difference because those cells cannot replace themselves. They do not have centrioles. So that's part of the reason why those injuries are so devastating, right? There's not going to be a huge amount of recovery after a head injury. Okay, that's depressing, isn't it? Okay. If you know somebody's had a stroke, that's very depressing, right? Knowing that things aren't going to get, they're going to get a little better, but they're not going to get hugely better. Okay. Okay, so those are the organelles, right? We've been through all the organelles, and I'm going to end up right back where we started with the nucleus. Just to come back here, the nucleus has that nuclear envelope around it. Remember, it has pores, it has openings in it. And that nuclear envelope is really a double membrane.
Okay, that's what I was gonna ask.
Double membrane, double membrane.
For the nuclear envelope. I couldn't really tell by looking at the cell.
I'm going to go way back up to this slide, Michelle. And this is where I first introduced the nucleus. And you can see that this is truly, there's a bilayer here. There's a bilayer, and here's a bilayer. So it truly is a double membrane. Okay? Okay, so you've got a double membrane. You've got 4 phospholipids, right? 4 layers, if you will, right? Because every membrane has 2 phospholipid layers. So you got double of that, and then you have those nuclear pores, these gaps or spaces in between.
Now, what is the outside of the nucleus?
The outside of the nucleus is now you are in the cytoplasm. So everything, so if I look at a picture, let me go down to where we were. Sorry, I'm getting dizzy too. Okay. Okay. So our nucleus, right, in the very center of the nucleus, what do you find? There's a dark staining center called the nucleolus, and also within the nucleus you would find the DNA, and the DNA wraps around proteins, and that whole thing we would call chromatin. Okay, so there's chromatin. Okay, chromatin. So what chromatin is what makes up chromosomes. It is going to be DNA wrapped around proteins. Let's look at a picture of this again. So here is the nucleus in my cartoon cell. The very center of it is the nucleolus. And out here in this area, all this is where the DNA would be hanging out. You can see little yellow ribosomes? Right? Those little ribosomes just kind of stuck on the outside of the nucleus. Those are not ones on the ER. Those are just ones kind of freely floating around. And you see those openings, those pores, right? There are nuclear pores. Then I want you to note that the membrane of the nucleus is continuous with the membrane of the ER. So this membrane, these cell membranes of the nucleus, of the ER, of the Golgi, and of the plasma membrane are always changing places, always moving around. Right?
They're all connected.
They're all connected, they're all ebbing and flowing. Endocytosis, exocytosis. And this idea of what's made in the ER blebs off and goes to the Golgi, and what comes off the Golgi blebs off and goes. And so there's a lot of these membranes moving throughout the cell, in and out. Watch this word with me. This one gives us some trouble. Nucleoli. This word nucleoli is plural for nucleoluses. Okay? So nucleoluses, right? Nucleoli. Versus more than one nucleus, nuclei. So be careful, those two words, right? Nucleoli, multiple nucleoluses, multiple nucleuses, right? Nuclei. Latin's a lot easier. It really is. Okay? So what do we find inside the nucleus? We find the nucleolus, multiple nucleoli, and it's here that we're going to be making ribosomal subunits. So what I want you to remember is that ribosomes are made, part of them, in the nucleolus. Ribosomes are pretty small. But where do we find ribosomes once they're made? Out in the cytoplasm. Look at those nuclear pores and look at the size of those ribosomes. Those little nuclear pores have to be big enough for the ribosomes to get out. Okay, that's one of the reasons that they're there. So there's a lot of information going in and out of the nucleus, but these little ribosomes, these yellow ribosomes, are partially made inside the cell and have to get out. So we have to have those nuclear pores to get those ribosome parts out.
Now the ribosome is going to be on the
Rough ER or just floating. Or just floating. Free ribosomes. You got it. Now, the DNA in the nucleus we know is called a chromosome. How many chromosomes do you and me have?
46.
We've got 46 chromosomes, right? We are human that's the way it is. Good Lord put 46 chromosomes in us. Other animals have a different number of chromosomes, right? Now, those chromosomes, what are they? 46. We have 46 very, very, very long molecules of DNA, right? Picture DNA, A, C, T's, and G's all strung together, double-stranded. And that long strand of DNA is going to be wrapped around some proteins. Together, the DNA plus the proteins make up a molecule called chromatin. Right? Chromatin is the stuff that makes up our chromosomes. And you only can see chromosomes during cell division. So during mitosis in lab, you're only going to visibly be able to see chromosomes under the microscope during active mitosis. During the rest of the cell cycle, you're not going to see chromosomes. You can see the nucleus, but you're not going to be able to see chromosomes inside the nucleus. So that brings me back to a picture, right, of DNA. This is my double-stranded DNA, right? And what's the outside of the DNA, I said before, was sugar phosphate backbone. sugar, phosphate, sugar, phosphate, sugar, phosphate. And what's holding hands here? Whoever asked, right? I've got, I may be wrong on this, I've got G's binding to C's, and I've got A's binding to T's. Okay. And that connection's always true. Now, if I take that double-stranded DNA, imagine up here at the very top of this picture, take that double-stranded DNA. Can you see it up there? Okay, now I'm going to twirl it around proteins. These proteins are called nucleosome. Okay, or the proteins make these little clusters called a nucleosome. So you got DNA wrapping around proteins, and then they wrap and wrap and wrap. And this is something most of you have never seen. This kind of looks like a telephone cord. You guys all seen telephone cords? I mean, I've had students who have never seen a telephone cord.
Oh, wow.
Right? We got to go to the Smithsonian to see those things. Right? But it kind of looks like a telephone cord, right? And so it's wrapping, 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 and then eventually what we see under the microscope looks like a letter X. Okay? That's kind of what we see for a chromosome. But keep in mind, what is a chromosome? It is the DNA wrapped around proteins. And if you have 46 chromosomes in your cells, that means you have 46 really, really long molecules of DNA wrapped around proteins. That's really what your chromosomes are, right? Very, very long molecules.
So there's only 46 cells that have them?
Nope, every one of your cells has 46 of these in the nucleus. Right? Every one of your cells, except egg and sperm, have 46 chromosomes inside the DNA, inside the nucleus. Okay. So that brings us to the end of this little section on organelles and cells, still in chapter 3. And so looking at this picture, you tell me, does this make sense to look at these common jobs? So the nucleus, right? It's okay to call it the control center. It's where the DNA is. The endoplasmic reticulum is where the proteins and the steroids are being made, kind of like on an assembly line. Now, once those things are made on the assembly line in the rough ER, where do they go? To the Golgi, right? And the Golgi is then going to be the distribution center. It's the Amazon, okay? So everything's being distributed from somewhere in Seattle or wherever, right? And the ribosomes were sort of the workbenches where many of those proteins were made on the assembly line. We've got the lysosomes, the cleaning crew for the company. We've got the security gate, right? The cell membrane, keeping the riffraff out of the company. We've got the mitochondria, basically the power generation. And we've got the structure, the infill structure, the building, if you will, made up by the cytoskeletal proteins. It's an okay way to look at it. Okay? So every cell is like its own little city, its own little village, its own little family. And you can kind of make stories. But if you look at these membranes, right? From the nuclear membrane through the ER, through the SER, through the Golgi, all that membrane blebs and moves back and forth along with the cell membrane.
So I have a question.
Yes?
It goes through the rough and then the smooth, and then
It goes rough or smooth.
So
It doesn't have, remember, because the rough ER is making proteins, the SER is making lipids, and so you don't necessarily have to go through both. Okay, but don't get caught up on that, right? But we're moving from the ER to the Golgi. Okay? Yep. We won't go there. We won't go that far. What do you think? Are we okay with the organelles?
Yep.
Okay. Click through them, be able to label them, know what they do. And that brings me now to the next part of this chapter. In the old book that we had, there's 2 chapters that were mixed together. And so I had to probably, this should be continuation of chapter 3. And I think your notes are correct, right? You don't have a break that says chapter 4, right? You just went right from that cell to this image? Right. I didn't even show this picture to you again, right? I just kept on going in your PowerPoint? Yeah, straight in, right? Yeah. So that's why there's a break there from a previous semester. So talk about chromosomes. No, this is still chapter 3 in Martini. The old book I used this is 4. I've got to do a little bit of reformatting for you. Okay, so we're talking about DNA. We just said that the DNA is, the chromosome is DNA wrapped around proteins. You've got 46 of these very long molecules in your nucleus of each of your cells. And we also stated, and you've seen it in the lab, you only can see chromosomes during mitosis. So when the cell is in G1 or S or G2 in the other part of the cell cycle, you're not able to see chromosomes under the microscope.
So G1 is not part of mitosis, right?
G1 is not part of mitosis, absolutely. So G1 and S and G2 are outside of mitosis, and you don't see the chromosomes at that point. Okay. Now in lab, last Friday, today, tonight, you're going to learn a little bit about the cell cycle very quickly, but the idea that every one of our cells is somewhere in its own cell cycle. And all of our cells are somewhere in this continuous, around and around we go. And we have to have new cells made all the time. I gave you a number a moment ago. I said about 25 million cells per second are produced. That's a lot of cells, right? It's a lot of cells being made and being lost. Now, most of those cells are now, they're part of your stomach lining. They're part of your skin, right? A lot of your blood cells are being made all the time and then broken down. So we're not aware of it, but it is truly happening. And I've already introduced this term to apoptosis, when cells die, because they're supposed to, right? This is apoptosis. If those cells weren't dying, then we'd be getting bigger and bigger and bigger, right? We just keep adding cells to us. Now, in the beginning, right? We'll start here and we'll finish up this story when we get to the very last chapter, when we get to the reproductive systems. But I want you to appreciate something. This is the female reproductive system. We're looking at an ovary. This is the fallopian tube, and there'll be another matching pair over here. And what's going to happen is that every other month or so, the ovary is going to release an egg. Ovulation occurs, and that egg's going to be sucked up in this little vacuum cleaner right here into the fallopian tube. If sperm are in this area, okay, and they have to be kind of way up here in this area for this to happen, then the egg will become fertilized. And that fertilized egg is called a zygote. Or egg and sperm will come together, we call that a zygote. And then the cell will undergo cleavage to 2 cells, 4 cells, 8 cells, and will eventually become a solid ball of cells. Here, I'm adding a stage. In lab, I just talked about the blastula, but there's actually a stage before that called the morula, and then the blastula. Regardless, you have a solid ball of cells here. It takes about 5 or 6 days for this journey to happen. So from the time egg and sperm meet here at the top of the fallopian tube, 5 or 6 days, you're floating down your mother's fallopian tube until you have reached this stage of development, the blastula, and now you are developed enough to grab onto the uterus to hang on for the next 9 months. Okay? Now, during this time, cells are growing very, very fast, right? And in fact, that's why in lab, we looked at a blastula. We looked at a fish blastula. Clearly, the same story is no different in fish. During those early days, the cells are growing very, very, very quickly. And we'll get into some more of these details when we get to the reproductive system. Now, it is possible. Sperm are viable for about 72 days. So sperm can live for about 3 days. The egg's only good for about 24 hours. So once the egg is released, it has to be fertilized within 24 hours or else the time's up. But that also means that sperm could have been there a couple days before ovulation or reach it within a day of ovulation. So there's about a 4-day window, right, for fertilization. And again, this is rapidly occurring. Now, it is possible for the egg not to get scooped up, and the egg is sitting out here in the abdominal pelvic cavity of the woman. And it's also possible for sperm to just keep on going and also get up in the abdominal pelvic cavity. So it's possible for fertilization to occur outside of the fallopian tube. And we call it an ectopic pregnancy. The woman would never know she was pregnant because out there in the abdominal pelvic cavity, there's no place for the embryo to hang in. It'll just die. It'll just die. It's also possible for fertilization to occur, and for whatever reason, it'll get stuck. The embryo will get stuck in here. It could be because there's scar tissue through here. It could be because, any number of reasons. But if the embryo gets stuck, that's also called an ectopic pregnancy. Now, in that situation, the woman will probably need to have an emergency procedure done because that embryo will try to grow, and it can actually start to push on and damage the fallopian tubes, and it usually has to be removed surgically. Either way, this embryo has no chance of surviving here in the fallopian tube.
Can those erupt in version?
Yep, it can rupture the fallopian tube and then make it more difficult for future pregnancies because you could have enough scar tissue in there. Yeah, so that early, it's going to happen pretty early. And I mean, the woman would not think she's pregnant in any way. It's very, very early in the stages, but a lot of abdominal cramping and pain, and it could be an ectopic pregnancy. Okay, so just a little bit about why do I tell you about the cell cycle? Because I think this is cool. I think the idea of thinking about egg and sperm and how rapidly they have to divide, and this whole story of the cells dividing 2, 4, 8, 16 cells, we realize how critical it is here in the early part of our development. Now, another place where mitosis is important, well, first of all, let's go through mitosis really, really quick. Okay? And I think this should be good. Now, some of you have watched the video, most of you have, I hope, and you have lab. The last group is tonight, right? Some of you have lab tonight. Anybody? Okay? You're going to finish up the story, see cells under the microscope undergoing mitosis. The rest of you have already been through this lab. So mitosis, literally, the word mitosis comes from the Greek meaning threads. Weird, right? Threads. When scientists first looked into the microscope and they saw cells undergoing mitosis, they saw these thread-like things. Well, what were they seeing? Chromosomes, right? They were seeing those chromosomes that look like threads. So that's where the word mitosis comes from. We can only see these, quote, threads during mitosis. And the 4 stages of mitosis, prophase, metaphase, anaphase, telophase. Right? And you should be able to recognize each of those stages. I'm not going to belabor that here right now. Okay? Say it again?
Is there a certain acronym to remember that?
Yeah, that will work right here down the bottom. I passed my anatomy test, right? So I, interphase. Now, interphase is not a part of this of mitosis, but it is a part of the whole cell cycle. So I, interphase, passed prophase, metaphase, anaphase, telophase. Right? I passed my anatomy test. And that might help you. That might help you. Now, this is showing the cell cycle. And in this particular artist rendition, they've really stretched out mitosis. And they're showing you what each of the cells look like in the different stages. So here's prophase, metaphase, where the chromosomes are lined up. Here's anaphase. The cell begins to elongate, and the chromosomes are being pulled apart. And then in telophase, you can clearly see cleavage, the cell becoming 2. And then there is a moment when the cell undergoing telophase splits, right? When it becomes 2 cells, at that very moment, we call that cytokinesis. Cytokinesis, cell, cyto, kinesis, movement. So the moment the cells are now 2 separate cells, we call that moment cytokinesis, and now each of those cells begins its own journey starting in G1. Okay, the first growth phase, then to S, and then to G2, and then back into division of mitosis.
So cytokinesis is not a part of mitosis.
Cytokinesis is not officially a stage of mitosis. It marks the end of mitosis. Okay? So mitosis ends with telophase, and then as the cells separate, that blessed moment is called cytokinesis, and now the cell is in G1.
I thought chromosomes can't do that,
Chromosomes can't do what?
the whole splitting.
They split during anaphase.
Okay.
So during this whole thing, you're going to see the chromosome lining up on metaphase. Okay? And then they're going to be pulled apart during anaphase, and they're going to have 2 sets of chromosomes going to each of the new daughter cells. Okay? So you'll pick up a little bit more about that in lab as well. Yes?
So basically, just like G1, are they just trying to make sure that the cell is working properly?
You got it. G1 is just when cells are doing what cells do. Growing, metabolizing, taking in energy, responding to its environment, doing all the things that cells do.
Okay.
That's good old G1. Then there comes a time where the cell goes into S phase. S for synthesis. And what it means is that at this point, the cell is going to plan for dividing. It's going to replicate or duplicate everything. It's going to replicate the DNA, make another copy of it. It's going to make sure there are plenty of lysosomes and peroxisomes and Golgi and everything else. And then it's going to go into a second growth phase, again, just kind of growing. So that means that when the cell goes into mitosis, it has already copied everything. So when it's going into mitosis, all it's going to do is very quickly split the pie, right? Everything's already been copied. I've already got 2 sets of all the DNA. I've got 2 sets of everything I would need, and I'm just going to very rapidly split the whole thing in half, through the process of mitosis, and then each cell starts off again on its own.
Is there certain ones that can't do that, certain cells?
Neurons. So the cells of your nervous system don't have centrioles. So they cannot, right? They don't have the centrioles. So they can't pull the chromosomes apart. So they can't divide. So that's a good example. Centrioles cannot divide. So they can't undergo mitosis. They can't replace themselves. Okay. When you're looking at these cells under the microscope, here's some idealized pictures. The first one here, this is just a cell, right? Boring cell. There's the nucleus. I can't see individual chromosomes. This cannot be a cell, therefore, in mitosis. We're going to say that this cell is somewhere out in the interphase. Interphase, let me go back. Interphase is all of this, from here to here. Interphase. It's the phase between the mitoses. It's everything between the cell divisions. So if I look at a cell and I can see a nucleus, but I cannot see individual chromosomes, I'm going to call that interphase. If I'm looking at the cell and I can start to see gibberish in here, right? There's some chromosomes showing up in here. They're still in a ball. I'm going to call this prophase. Yep. If I see the chromosomes and they're all lined up in a row, metaphase. If I see the cell elongating and I can see the chromosomes being pulled apart, that's anaphase. And then if I see cleavage, clearly the cell is becoming two, and I see the chromosomes are separated, that is going to be telophase. Okay. Now what else is happening? Back in prophase, the nucleus is disintegrating. So the nucleus is going away. The mitotic spindles are forming. See all these things are starting to go out? Mitotic spindles are forming, and the chromosomes become visible to me. And when I get over to telophase, the exact opposite happens. The nucleus is going to reform in the daughter cells. The centrioles are going to break down. The mitotic spindles are going to break down. And the chromosomes are once again going to become invisible to me under the light microscope. So everything that happens in prophase is reversed in telophase. Pretty cool stuff, right? I know it's kind of academic. And I know that somewhere along the way you've learned about mitosis and you're like, oh, here we go again. But it really is important to understand that this process is happening so very quickly as we think about the cell cycle. I not only want you to get excited about the cell cycle because of early development, I also want you in a moment to get excited about some other devastating thing that happens in many of our lives, and that's cancer. Because I'm going to walk into cancer here in a second, and what is cancer? A rapidly growing cell that's going out of control. So we need to understand mitosis if we want to ever understand cancer. Before I get there, though, there's two types of cell division going on in your body. The first is what I've been describing, and it's really the only one you're going to be dealing with in great detail, and that's mitosis. This is when the cell is actually making another exact copy of itself. There is, however, another type of cell division occurring in your body called meiosis or meiosis. Okay? And meiosis is only occurring in your sex cells. That is, it's only happening in the formation of sperm and the formation of eggs. And that is where meiosis occurs, and only there in your body. Here's the deal with meiosis, just to get an appreciation of it. Meiosis is not going to make an exact copy. Meiosis is used to create egg or sperm, and each of these cells, each egg or sperm, is going to be a genetically different a variety of the parent. This whole process of making egg or sperm is referred to as gametogenesis. Gametes, another word for the sex cells, egg and sperm. Genesis, in the beginning. Okay, so the production of gametes requires this process called meiosis. And here's why we care, right? Anybody in the room recognize the condition this young lady has? Down syndrome, right? And Down syndrome is because she was born with an extra 21st a problem in meiosis. Okay, so I just want you to appreciate that this may seem really academic, right? But when meiosis doesn't go right, you can end up with a child with Down syndrome. So here's a cell, okay? And what happens in meiosis is that the cell divides twice. It doesn't divide just once like mitosis, it divides twice. So it divides once, the chromosome should be spread to each side, and then it divides again. What happens in the formation of Down syndrome is that some of the chromosomes don't separate like the way they should. Okay? So rather than getting a cell with one of each chromosome, look what happened here. Both of these stuck over on the left. Okay? We're not going to get too fancy about this, but we see that an extra chromosome went over here. They didn't separate like they should have, and you have both pairs. Then when this divides again, both of those end up in this particular gamete. Now, if this is the egg or the sperm that is going to create this young child, then every cell in her body would have what we call trisomy 21, a third copy, tri, of the 21st chromosome, and that causes Down syndrome. If instead this egg or sperm had been used, there would be no baby. Right? This is a non-viable lethal. This sperm or egg doesn't have any chromosome 21, and that's not consistent with life. So you can have an extra chromosome, but you can never be missing a chromosome and still be normal.
What's the other side then?
Other side is normal.
Okay.
Yeah. So in this case, only half of the egg or sperm would be a problem. One forming Down syndrome, one forming DUD, and then these would be normal. Okay? So again, this is definitely a process that goes wrong sometimes, and we know it goes wrong more in older women, that eggs are stored. We'll talk about this when we get to the reproductive system. But eggs are stored. Eggs are already in every woman before she's born. Okay? And she stores them for 20, 30, 40, 50 years. That's very different from guys, right? Guys are not making sperm until puberty, and then they're making it fresh loads every day. And so sperm never get old, but eggs get old. And so when the eggs are stored, they are more likely over time to have these non-disjunction, these separation issues. And so with increasing maternal age, you're going to have an increase in Down syndrome. Okay?
I had a question too, because some kids I've seen look like they got Down syndrome, like a close call, or because you can look at their face, but they're not a Down syndrome child.
No. Okay.
I'm like,
Now there is a look to Down syndrome, right? There is a look, and that look is all races, right? You'll see a black, an Asian, whatever child with Down syndrome, they have that same similar look, and those looks are, well, their eyes are separated. They've got heart defects. They've got some mental retardation. There's a lot of things that can be wrong. Now, there are some milder versions of Down syndrome. I'm not going to get into that, but about 5% of Down syndrome kids are not, quote, full-blown trisomy 21, and they've got a less severe case of it. Becky on Glee.
Oh, on Glee, okay.
Very high functioning.
What was that old show?
Very likely. What was the name? Corky.
oh yeah, Corky.
Okay? Corky. Life goes on. Okay, so these are some individuals who clearly were able to do a lot more than the average individual with Down syndrome. I would argue they're in that 5% range where they have a milder case of it. They're not as severely affected intellectually.
How does that happen with that? That thing, like either they get it or they don't.
How does what happen?
When that thing, they say
Yeah, if the chromosome down here, if this is the egg or sperm that's used because there was a problem, then every cell in this individual would have an extra 21st chromosome, and they would have Down syndrome. Okay?
how do they get to the borderline? That's what I understand.
How do they get? Oh, let's not go there.
Oh, okay.
There's another way that this can go wrong, and gives you a more mild case of it. We're just not going to go there. My point going here was simply to say, you know what? Meiosis seems really academic, but look, we probably know somebody or know a family member, a cousin, or somebody down the family tree where someone in the neighborhood has Down syndrome. We now recognize that it's because meiosis went wrong, right? There are reasons for us to learn these things. Yes, ma'am.
Can it happen like an equal chance of whatever chromosome?
It doesn't have to be 21. You could have trisomy 18, the 18th chromosome. The thing is this, there's only 3 trisomies that are able to survive. Okay, Down syndrome is the most likely trisomy to survive. This could happen with chromosome number 20, and they don't survive. Chromosome 22, they don't survive. There's only 3 different chromosomes that can be in triplicate where they survive. Down syndrome is the most forgiving. You can live a reasonably normal life, right, with Down syndrome. Trisomy 18, trisomy 13, those kids are severely affected and don't live very long. Any other trisomy, lethal. They never get out of the womb. They would never be born.
And the bottom one with those 3
Trisomy just means that
divided into 3?
trisomy just means there's 3 copies, right? There'd be 3 copies.
So if you're talking about going down 3 times or what?
Yep. So there's only 3 different kinds of trisomies that are viable. The rest are all lethal. They would never make it out of the womb. Okay. Okay. So again, another example. I think I can hopefully get you a little more excited about cell cycle. I mentioned apoptosis. Here is human from week 5, 6, 7, 8, 9, 10. And during these 5 weeks of development, the fingers and toes are being formed. At week 5, the arm hasn't really protruded at all, and the hand is more of a paddle. The fingers have not yet separated. And over the next 5 weeks, cells are going to die in that paddle to individually form what will become the fingers. Right? So the fingers don't sprout out as 4 different things. They actually start off as a paddle, and then cells in between them die off. They know to die, right? Apoptosis. And by dying at the right time in the right place, now you create fingers. Now some people are born with extra or webbed fingers. What didn't go right? Apoptosis didn't go right, right? Something didn't happen during development. But here's week 10, right? Here's a human finger and thumb, adult, and little toes from a baby, about 10 weeks old, in utero. And then you can see the fingers totally formed, right? By the 10th week. So by 2.5 months, the limbs are formed. Okay, so now why else do we care about cell cycle? This ugly thing, right? Tumors. Tumors are rapidly growing cells that are out of control. Normally, cells know when to grow and when not to grow. They know when to divide and they know when to die. Right? They know when to apoptose. The problem is when that balance becomes out of control and cells start growing faster than they're dying, that cell can become a solid tumor, a mass. A tumor, neoplasm, another word for it, new growth. Okay, so you got a solid tumor. Potentially, there's other kinds of cancers, but this is a solid tumor kind of cancer. So you get this ball of cells that are growing faster than they should. I'm going to break this up into 2 basic groups. Now there are dozens, hundreds of different kinds of cancers, but I'm just going to break it up very simply. There's benign and malignant. Benign neoplasms, benign tumors, here's the deal. They usually are more slowly growing, okay? And I told you Benny means good, so if you have a benign tumor, this is the better of the 2. Okay, pray for a benign tumor. Here's what happens. Any tumor, the cells de-differentiate. Now that's a big word. Basically it says this. When we're back in the blastula, right, that little ball of cells, those cells haven't yet gotten their instructions. They don't know if they're going to be muscle or kidney or liver or brain, right? They don't know what they're going to be. But as they do become different, we say the cells have become differentiated. So as development goes on, some cells become skin, some become liver, some become kidney. In tumors, the cells are usually de-differentiated. It usually means they kind of go backwards to a more adolescent or childlike state. Well, what do childlike cells do? Grow faster, right? We know that cells in the embryo, cells in a young being grow faster. So these cells de-differentiate, they become less specialized, they find a way to get their own blood supply, right? Because a tumor, right, it's a solid ball of cells. Every cell in the tumor must also have blood and oxygen and nutrients. So if we don't get blood supply to a tumor, then the tumor can't grow. So tumors have their own little tricks to get more blood supply to them. Hold on. And these tumors are usually not lethal. These are slower growing, unless this tumor is deep in the brain and they can't operate on it, or unless this tumor is pushing up on a major blood vessel, pushing up on the airway, pushing up on something. These typically, as long as they can be removed, are not going to be lethal. We can typically take a melon scraper basically and pull them out. They also stay confined on the top. They stay within a connective tissue bubble, so they don't spread, usually. Benign tumors do not spread, slower growing, but still have to have their own blood supply. Question?
So, okay, that's a benign tumor. I was just thinking when you said it needs its own oxygen, right?
Yep.
So I'm thinking that it clings on to something.
It does.
Yeah.
They're actually going to cling on to something and hold on to something, absolutely. Because every cancer is one cell in your body going nuts. Right? Cancer is one cell out of your 75 trillion cells going crazy.
Wow, that's
A tumor is it's a clone, right? The whole tumor, every cell that's in your body that's cancerous is of the same cell out of control.
Now, I don't know, my mom expressed to me that she was a twin, but the tumor had died off. And
Yes.
when they removed it, it had like hair.
Hair and fingernails and teeth. Yeah, so it's possible you can have what's called, there's another name before it, it also has the word mole in it. And basically, sometimes a fetus, something goes wrong in the development, and what is created is this ball of hair, skin, teeth, bones. It looks like one of those little Furbies that went wrong. Okay? No. And sometimes there can be twins, and one twin is fine and one twin's a mole. And there's another name in front of it, I can't remember right now. But nothing develops correctly, right? And they have these little Furbies. Looks like Furbies with teeth. Yeah.
so you say it's one cell going bad, then does it attract other cells or something?
Oh, no. Yep. So one cell's going bad in a cancer. Now, the other kind of cancer is malignant, right? A malignant cancer. Now, a malignant neoplasm is the one you don't want to hear about. These are unencapsulated. They don't stay in their place. They tend to travel, to metastasize. They travel around. That's the problem. They too are dedifferentiated. They go back to an earlier state and are faster growing. They also increase their vascular blood supply, but they're growing very rapidly, and here's the kicker, they spread. And they're going to spread very easily through the blood or through the lymph, through the inner lymphatic vessels. Okay? And this is what's dangerous, because it could be a brain cell that's gone cancerous, but that brain cell will migrate and go to the bone, or to the lung, or to the kidneys, and set up shop there and now form another tumor. But when they do the biopsy, they're going to be able to tell where that cell came from. So when they do the biopsy, let's say there's a cancer of the kidney, and that cancer spreads to the bone. When they take the cancer from the bone, they're going to be able to tell it's really a kidney cell that went nuts, that's in the wrong place, right? So that's what cancer is, ugly, ugly stuff. It just takes one cell. Now, what does a cancer cell look like to you? When you think of cancer, what comes to mind? Ugly, black, ugly thing. They're beautiful. Because what are they? Rapidly growing, young, not healthy, but rapidly growing cells, right? So when a tumor is taken out, or when a mass of cells is taken out that's cancerous, they're not black and ugly. They're typically beautiful looking cells, right? They're just in the wrong place and they're out of control. Okay? Oh, yeah, another way of thinking about it. So cancer, here's the whole idea. Cancer cells lose control of their cell cycle. G1, S, G2, mitosis, G1, S, G2, mitosis. Something goes wrong. Another thing is that these cancer cells lose contact inhibition. Let me try to describe this to you. If I took some of your skin cells right now and I put them into a petri dish with the right environment and the right nutrients, your cells would grow on the bottom of that petri dish, and when they got to the edge, they'd stop. When they touched another cell, they would stop. Most cells know that when they come up to another cell, that's where my property ends, right? And they're good neighbors and they stop spreading. Cancer cells lose the rules, and they lose that contact inhibition, and they start rolling over each other, and they start making a solid ball. And so they no longer are inhibited by space, okay? Most cells would stop growing when they touch each other. So these cancer cells will start growing over on top of each other. Again, cancer cells will de-differentiate back to an earlier state, and they're going to get more blood supply. The term for this is angiogenesis. We know what angio means. Angio, vessel, genesis, in the beginning, the formation of. So angiogenesis, the formation of new blood vessels. So tumors are going to undergo a lot of angiogenesis, and these cells, not only will they spread, but they'll squeeze in places they normally wouldn't go. They literally will squeeze and go, to other parts of the body. And that is called metastasis, or the cell has metastasized. Okay?
Spread?
Metastasized. What's that?
When they spread?
When they spread, right? When they spread, when a malignant tumor spreads, we call it metastasis, or the cell has metastasized. Okay, question?
So benign neoplasm cells, they're still considered cancer cells,
They're still
right?
cancer. They're still out of control. They're going to grow a little bit more slowly,
Yeah,
and they're not going to spread.
okay.
Okay, once a cell breaks out of its capsule and spreads, it's no longer benign.
No, it's malignant.
Now we're calling it malignant, right?
Benign's still pretty bad.
So benign, if you get a diagnosis of a tumor, pray for benign, right? And that it's in a place where we can grab it.
Cause I've
Okay.
had like tumors on my hands right here, you can see it like a cyst. they
Right,
say.
right,
Yeah, but it wasn't malignant, benign.
right. Just take it and watch it, and yeah, everything's fine. Yep. Okay. Tell you what, guys, this is a good place for me to tell you about your next exam, just to give you a view of what's coming up. And I need to upgrade those pictures, okay, or those pages. So ignore the pages and ignore next Thursday. Forget all that stuff. But I do want you to look at these words. On your second exam, there will be a short writing assignment. You're going to know the writing assignment in advance. And here's basically what it is. There are 11 terms here.
Yeah, it does that.
You don't have these?
No, we don't
Yeah,
have the
we have
first
the
two
things.
bubbles.
Took all that off?
Yeah.
Yeah.
I was good. Okay. I got to clean up my own copy.
Yeah.
Your copy's better. Here are the terms. Some of these you know, some of these you've never heard of yet, perhaps. Transcription, nucleus, DNA, translation, proteome, cytoplasm, ribosomes, proteins, rough ER, genetic code, and mRNA. How many of those words have you already heard of? We've already heard of nucleus, DNA, cytoplasm, ribosomes, proteins, rough ER, right? That's what we've heard of. The other ones are coming in the rest of this chapter. And what you will be doing is on the second exam, you're not thinking about it now, I know, but on the second exam, you will write 3 sentences. And in those 3 sentences, you must include at least 10 of these 11 terms. Okay? And you got to make sense. So the sentences have to make sense. It's not that difficult. Okay? But if you can write a sentence and use these words in it, that means that each of your sentences will have between 3 and 4 words in it. This is sort of like when you're reading the back of the chapter and they have that nice little synopsis, that nice little review, right? And they have those nice sentences, but they're all highlighted with all the key words. You're kind of writing that. You're using these words in 3 sentences.
You can only use 3 sentences?
3, you can use 2, right? But I don't want any more than 3 sentences. What I definitely don't want is 10 definitions, right? And the question is always, well, Dr. Macauley, what is a sentence? Right? I'm not kidding you. What is a sentence? When I grade these, I'm looking for 3 capital letters.
How many commas?
Right? Capital letters. And a sentence ends with a period. I'm counting capital letters and periods. I'm not going to take you off for run-ons. I don't care, right? If it runs on a little bit, okay, comma, whatever, I'm not going to get upset about it. But these 3 sentences need to use at least 10 of these words. When we're done next time, now, I won't be lecturing until next Monday. On Wednesday, we're just coming together, having fun, having a test. Next Monday, I'll continue this conversation. We should finish this chapter up next Monday, and then I'll tell you more about this little assignment. But I'll let you out about 5 minutes early today because I'm feeling generous, and it's a good place to stop. And how am I doing on the syllabus? Who has their syllabus in front of them? Am I on track?
Yes.
You are.
I am. And next Monday, what should I be doing?
Finishing chapter 3.
Perfect. Okay, I'm right on. Okay.
Chapter 4
There we go.
Wednesday.
Perfect. Absolutely. So we're on track. I'll see you all on Wednesday for exam number 1.